So at this point, we've taken a look at glycolysis, the Krebs cycle, and what have we gotten? So far, we've got a little bit of energy from glycolysis, but preparation process. We got a little bit of NAD charged up in glycolysis. Krebs cycle didn't get as much ATP, but charged up some of our electron carrier molecules. And that's necessary because that's going to be the big push in this next portion. Don't forget the main job of the citric acid cycle, the Krebs cycle, was to rip the carbons uh, from that glucose and get rid of it so that we could breathe it out. But notice now we haven't produced a lot of ATP, and it takes a great deal of ATP to run a living cell. So now we have built up the energy carrying molecules necessary, the NADH, the FADH2. We're going to use those now to truly produce a chunk of ATP and power a cell. This goes through what's referred to as the electron transport chain. The electron transport chain happens via enzymatic molecules that are embedded in the inner mitochondrial membrane, okay? And happens on a level that's referred to as oxidative phosphorylation now. Okay, back in glycolysis, it was substrate level phosphorylation, but now we're doing phosphorylation on an oxidative level. Okay, each step in this process. Okay, so on the y-axis over here, you can see free energy. Okay, relative to oxygen. So here's our Gibbs free energy, our delta G, if you will. Okay, notice each step in this process is a cascade of free energy down, okay, down the free energy slope here. Notice the NADH comes in at the top, okay, the highest energy. The FADH2 comes in a couple steps in, so it's not quite as high energy as the NADH. But all of these now can go through that energy coupling, driving and moving our electrons from one energy level to the next. And in the process, each piece of energy that's ripped here, okay, can be used. We'll show you in just a second how that's going to be used by the cell. Okay, notice the very last electron acceptor here is oxygen. Okay, the very last electron acceptor in the electron transport chain is oxygen. Okay, so the energy released here is going to form an electrochemical gradient. Okay, a gradient just like having a great deal of any particular chemical in one spot. Okay, the glucose that originally diffused into our cell had a gradient of high outside the cell, low inside the cell. Now we're going to have an electrochemical gradient, a gradient of ions, and that's going to happen across the inner mitochondrial membrane. Let's take just a second to review the anatomy of this mitochondria. So here we've got two membranes. The outer one is smooth. The inner one is highly convoluted or highly folded. And what we have is the necessary enzymes for this electron transport chain are embedded in this inner mitochondrial membrane. Okay? So why do we need this to be folded? Remember, when Ma Nature needs a lot of space in a small place, she's going to fold it, she's going to stack it, she's going to twist it. Okay, so we need a lot of this inner mitochondrial membrane because that's where these enzymes are embedded. Okay? So that which we've discussed, the NAD being now oxidized, okay, and moving from one step down that energy level of the electron transport chain, okay, is flowing through these different enzymes embedded in the inner mitochondrial membrane. Okay, so notice the colors here match the colors down here. So the dark tan is the inner membrane space. Okay, we have our membrane itself, and the light tan, okay, is the mitochondrial matrix deep in the inside. So the colors here match the colors in this close-up diagram. Okay. At the tail end of this, here's the end of the electron transport chain. We have oxygen taking that final electron, being that final electron acceptor, forms a molecule of water.